Good afternoon, everyone. Pleasure to be here today and a privilege to present on behalf of Atara Bio and the team what type of progress have we made over the last few months since last time we presented the company to you a year ago. I mean, Atara is, after the usual forward-looking statement, which is, heard by now, is really dedicated to patients in many ways. I mean, here we have as a mission to transform the life of this patient. And each and every of these patients on this slide have come to our company, have talked to our people, to everybody in the staff, to be able for everybody in the company to understand what have they been through, the journey, the type of life that has been their life, and the issue that they had with treatment, therapies, and the burden of their disease. And we believe it's very important to have this continuous dialogue with patients, to be including that into everyday work that we do at Atara for them, on their behalf, to transform their life. So that's really what we call patient focus. And that allows then the people, whether they work in supply chain or in clinical development or in finance, to really know why they are working every day. So many of them are not coming to the office anymore post-pandemic, but at least they know why they are waking up and putting on their computers and working every day. And we are really working for these patients. Now, the way we try to transform the life of these patients is through a very differentiated type of platform, relying on Epstein-Barr virus T cells that are being transformed and manufactured to be able to create new therapies for various diseases. We have two therapies in the clinic right now, Tab-cel and ATA188, more about that later. And we have a number of pipeline assets that are in late preclinical in terms of allogeneic CAR T is going to come to the clinic very soon. What is unique about our platform is really the way we are taking cells from healthy donors, being able to make EBV T cells that are very specific EBV T cells that are really designed to be able to address different types of EBV, Epstein-Barr virus associated disease. Either some type of EBV-associated cancer. As you may know, EBV as a virus is the first oncogenic virus. It's associated with many types of cancers, both liquid and solid tumors. So we are addressing these specific cancers with products that are designed to address the different type of antigens that are being found in these cancers. And then we have another type of product like ATA188 that is designed specifically to address some autoimmune disease that are caused by EBV, like multiple sclerosis. Now, that's only the first part of our pipeline and our activity, because what we can do is to further transform these cells and in inserting chimeric antigen receptors to then leverage natural biology of EBV T cells, but to give them another target than the EBV and then endogenous TCR, which is based on the chimeric antigen receptors, and we are developing both mesothelin CAR Ts as well as CD19 and other type of targets for CAR Ts. So same platform, either addressing EBV-associated disease or addressing many types of solid and liquid tumors with chimeric antigen receptors and creating true allogeneic CAR Ts. Now, a few words about our first product, because that's a very exciting time for us right now. We are really very close to get the first approval of the first ever allogeneic T-cell therapy approved in the world, and that's coming in Europe in the next few months. We are strongly convinced that this is going to be a landmark for us as a company, but more importantly for the patients, because they are going to get access to that therapy where today their prognosis is very poor. Now, the product is addressing, in its first indication, a particular disease, which is a rare case of lymphoma occurring in patients that have gone through a transplant, either solid organ transplant or HCT transplant. And this particular rare case of lymphoma is extremely serious and of poor prognosis, because patients have a median life expectancy of just a few weeks to a few months. And by 2 years, most of them are dead. So here we are with a product that allows in the pivotal study to have up to 50% response rate. And once the patient is responding, they survive. In fact, the level of survival at 1 year is quite spectacular in this first study, around the 80% level and over. Now, not only in the short term over 1 year, but we have long-term survival data, because what we see from previous studies with Tab-cel is that when you look up to 5 years following treatment, you have a very high level of survival, either in patients having complete response or even in patients having partial response, which is quite unusual in lymphoma. So whether you have a CR or a PR as a patient, you are able with the treatment and with favorable safety to be able to survive for a long time. So this is very close, hopefully, to be talking about cure at one stage. Still have to wait a few more years, but we are on the path to have a cure for these patients. And that's amazing because these prognosis, the prognosis of this patient is, as I say, very poor. So we offer them hope, and in half of them, the ability to survive long term. With, at the same time, a very favorable safety and no major issues, whether it's from a CRS point of view or neurotox or others. Now, the second product we're developing, and that's another very exciting endeavor, it's probably the boldest type of bet we're making right now, is to develop a treatment for multiple sclerosis and other autoimmune disease based on cell therapy. We have with AT188, and we have 2 fast-track designations in the US, a very unique therapy from that point of view. Now, what we're trying to do is really to address what you know is a debilitating disease, MS, where there is no treatment today able to reverse the progression of the disease. All the treatments that have been approved and even are being developed today, apart from AT188, are just trying to delay the progression of the disease by a few months. Here we're talking about a treatment that has the potential, we believe, to reverse disability progression, to improve the patients in terms of their disability. This product is very specifically designed to address what we believe is at the core of the disease, and we believe that because there have been mounting scientific evidence to show that MS, multiple sclerosis, is in fact caused by infection with Epstein-Barr virus, and it's a latent infection that is causing, through different type of mechanism, the triggering of the onset of MS and then the progression of the disease and progression of the symptoms there. These 2 papers, on the right you have the Science paper that was published in January, and then the Nature paper on the left that was published in February or late January as well. These 2 papers are really landmark papers to show that, on one hand, MS is really caused by EBV, and following more than 10 million military personnel, a great team in Harvard have been able to show that you need to start with an EBV seroconversions, then you start to see axonal damage, then you start to see the symptoms of MS. And the other paper is showing how that could be possible. And one of the core explanations could be very well that you have molecular mimicry. That means that some of the part of the EBV protein are very much alike some brain protein. So there is mistaken identity here for the immune system. It's starting to attack the brain protein instead of attacking the EBV-infected cells. So on that basis, we've developed AT188, and it's really a product that is specifically designed. It's a targeted therapy for MS to eliminate the B cells and the plasma cells that are infected by EBV and are immortalized in some compartment of the brain, and therefore are able to permanently stimulate the autoreactivity that is leading to the progression of the disease in the patients. Now, in our first human study, in 24 patients, small number, but we saw some interesting and encouraging data in the sense that we had a number of patients improving and then a large number of patients not declining, which is quite interesting in that type of disease because by definition, progressive MS means that the patients are progressing and declining. Now, this was real in terms of the benefit. I mean, you have on this slide the type of benefit that the patient occurred, and it's not short-term benefit. Some of these patients, if you take patient G or patient C, were able to improve significantly their mobility. They were having the requirement of a walker to walk, and then suddenly they can walk without a cane for 200, 500 meters. So that's a profound change in their mobility that is, we believe, a sign of the efficacy potentially of that product in the brain. And why do we believe that's happening? We believe that's happening because we've also saw sign of remyelination in the brain of this patient. In what is called the enhancing T2 lesion, the chronic lesion of the brain in MS patients, we've seen with biomarker in the patient that have improved under AT188, remyelination occurring. So that's why there seems to be something happening there that is quite exciting. Of course, this is an open study, small numbers, no evidence yet. But we are working now on a randomized double-blind study, and that is going to read out at the end of next year. It's going to be a very exciting moment for the field to be able to bring cell therapy to patients, a very large number of patients with multiple sclerosis, with the hope to change their life forever. Now, the last part of our portfolio activity is really around CAR-T and creating allogenic CAR-Ts, leveraging against these EBV T cells, natural biology, maintaining the TCR, the endogenous TCR, which we believe is a key survival signal for the T cells. So therefore, increasing the persistence and the functional persistence of this allo CAR-T. So we have an EBV T cells, we insert chimeric antigen receptors, and then we give another target to that cell. And in doing so, maintaining the endogenous TCR using next-gen technology in CAR-T, we are able, and we've seen a lot of preclinical data already, to enhance the functional persistence of the cells. So they will be able to better traffic, better penetrate, and persist, even in solid tumors. And we add things like PD-1 dominant negative receptors, for example, in solid tumors, to be able for the cell, the CAR-T, to resist to the immunosuppressive environment that they encounter in solid tumors. Now, first, most advanced product is a 3219, which is a CD19 allogeneic CAR-T, developed as a potential best-in-class, but also as a proof of concept of the platform. It's going to the clinic next year, but it's supported by early clinical data that were coming from Memorial Sloan Kettering in New York, where they show that in this type of construct, they achieve a high level of CR, of complete remission in this patient, up to 83%, with very long-term durability of remission, more than 2 years. Again, that's arguably the longest durability of remission seen with allogeneic CAR-T so far, and we hope that we can reproduce this type of data with our product is going to the clinic very soon. So all in all, very active portfolio of activities, again, relying on the same platform, allogeneic EBV-T cells, either fully transformed to have another target with CARs or just focusing on EBV. And we have a number of programs going to read out over the next 12 to 18 months with very exciting time ahead for us and hopefully, and more importantly, for the patients. Thank you very much for your attention. Look forward to talk with you later.